Hey guys, Mr. Hyatt here. This is part 1 of the AP Biology Chapter 45 lecture on hormones and the endocrine system. So hormones are going to be involved, specifically in animals, are going to be really involved in regulating lots of different processes. We're going to look at several examples of antagonistic relationships where one hormone causes something to increase, one hormone causes something to decrease. So should be lots of opportunities to look at negative feedback here in this chapter. So just kind of keep that in mind as you move through the unit, through the lecture. I apologize. So hormones are going to be broadcast to all parts of the body, but only the target cells are equipped to respond. So sort of like the idea that radio signals are broadcast across the world, but only if you have the right device can you tune into that signal. You can't just raise your hand and open your mouth and have the radio play out your mouth. You have to have the right device to receive that signal. Insect metamorphosis and many other processes are regulated by hormones. Plants have hormones. We'll talk about those specifically with pheromones later in one of the following lectures. So remember from our nervous system lecture that we've got two communication systems. Nervous system is going to move high-speed signals, get things long distances, but we're going to have to relay them, kind of like playing telephone, through those synaptic messages that we covered in our last unit. Endocrine system, they're going to be longer acting responses, things like metabolism, things like development. Those aren't, hey, you got to get your hand off that hot stove before you melt your skin off. If it takes a little bit longer, okay. So that's where the endocrine system, that's where we utilize those. Hormones, in our last unit we talked about ligands. Hormones are going to be our ligands in our endocrine system. So they're going to be passed from the cell that creates the signal to the target cell. They're going to connect, they're going to find a receptor, and they're going to enter the cell or cause a response by the cell based upon the receptor and specificity and all those types of things. We've got a couple of different signaling molecules or a couple of different ligands. Each of these gets its own slide, so won't spend a lot of time on it there. But before we get into that, we got to figure out what is a hormone. Hormones are chemicals that are going to be excreted into the extracellular fluids. They're going to travel through the bloodstream throughout the whole body. We've got endocrine glands and exocrine glands. Essentially, the difference is how specific are you delivering your message? How specifically are you delivering your message? An endocrine gland, got a picture here. This is a good example of an endocrine gland. I'm producing this hormone and I'm releasing it. Diffusion will allow diffusion will allow the hormone to enter a blood vessel, high concentration to low concentration. Then that blood vessel is going to carry the message somewhere, and when we get to, really once we clear this area, we're going to have more molecules inside the blood vessel than we have outside, so the molecules, the hormones, will diffuse out of the blood vessel. So that's an endocrine gland. You see there some of the things that hormones are going to mediate, again, controlling slow processes. Exocrine glands are similar, but they've got a duct that carries the hormone someplace specific. Tear ducts are an example. You don't cry through your whole eyes. You cry out your tear ducts, and then the water goes wherever it goes. Moving on to local regulators, they're going to be chemical signals that travel only a short distance. That's why they're called local regulators. Paracrine signaling and autocrine signaling are kind of the two specifics. They're going to control things like blood pressure. They're going to definitely think about those synapse, the neurotransmitters. Those are our local regulators. Paracrine signaling, that's sort of like radio. Make the signal, broadcast it. Anybody that's around you, you turn your music up really loud at home, all your neighbors hear it. That's paracrine signaling. Autocrine signaling, same thing, but you're trying to deliver the message to yourself. There are a few examples of that in the human body. Not a ton. Neurotransmitters and neurohormones, we know about these. We don't have to spend a lot of time on these because we covered that in our neurons unit. They're the chemical signals that are going to go from one end of the synapse to the other, take it from the axon to the dendrite of the next neuron. And, of course, we know lots about that. Neuro, that should scream brain. So a neurohormone is going to be a specific kind of a hormone that we make in the brain, diffused into the bloodstream, and it gets sent throughout the body. Endorphins are a really good example of that. We'll talk more about endorphins later on. Here's neuroendocrine signaling. Again, we've got this secretory cell. Again, it's a nervous cell that's causing this molecule to be created. The axon releases these neurohormones. In this case, they're hormones. They're not neurotransmitters. And they're going to travel, into the bloodstream and be carried throughout the body. That's a little different than what we talked about with neuronal signaling, where this signal goes from one axon to another dendrite. In this case, the axon is connecting to the blood vessel so that we can export those hormones throughout the body. Pheromones are chemicals that are released from the body. You're using your hormones to communicate. Notice that they're outside the body. They're essentially hormones that are outside the body. If you've ever seen ants following one another, playing that follow the leader kind of thing that they do, they're following pheromones. They're following the trail of the ant in front of them. Sometimes that's used to attract potential mates. Sometimes it's used to warn of predators. Like if certain species are killed, they'll release a pheromone, and that tells other individuals in the area that, hey, stay away from this. This is something bad's going down over here. We've got three different chemical classes, three different types of molecules that are going to function as hormones here in invertebrates. Polypeptides, notice those are proteins. Amines derived from amino acids. So that's kind of protein-based, maybe a little nucleic acid-based, depending on where we're talking about the human body. And then steroid hormones are going to be lipids. So the solubility of the hormone is going to be huge. It's going to basically dictate whether we're going to have a signal transduction pathway to get from the cell membrane to the nucleus, going through the cytoplasm, or if the ligand can diffuse into the membrane, through the membrane, into the cell, and can physically alter transcription factors, translation, something like that, inside the nucleus. So a lipid-soluble hormone that's a steroid passes easily through the cell membrane, while water-soluble ones, the polypeptides and the amines, don't. So that's going to be, logistically, that's going to be very different. Here are a couple of examples. Left side, those are our water-soluble hormones. On the right side, those are our lipid-soluble. So these guys are going to go straight into the nucleus and alter translation. These guys are going to be received at the cell membrane, and some messengers are going to have to carry the message to the nucleus. Pay special attention anytime you see thyroxine. That's one that's really heavily tested over on the AP exam, not specific, well, also on my unit test, but it's on my unit test a lot because it's on the AP exam a lot. So anytime you see thyroxine, pay attention. So, like I said, based upon where the molecule can go, our response pathway is going to be a little bit different. Kind of already summarized that stuff, so go back and re-listen to it if that didn't make sense. Of course, cell signaling, we've got reception, transduction, response. There's a kind of an outline of reception. So we excrete this hormone. It's water-soluble. So since it's water-soluble, it's going to diffuse through the bloodstream. Then it's going to bind at the target cell on a receptor. Fat-soluble, all this is the same, but then it's going to dissolve through the membrane into the nucleus and find a signal receptor. We continue that process out. We've got to have a signal transduction pathway to get a cytoplasmic response or a different gene expressed. Over here, fat-soluble, that ligand comes into the nucleus, binds to a receptor that causes a change in the genetics, causes something to be produced that wouldn't normally be produced, and that produces a cytoplasmic response. So just different mechanisms, different ways to get around solubility, essentially. So the hormone epinephrine is a water-soluble hormone that's it's an EpiPen. If you've ever heard of an EpiPen, that's injecting epinephrine. It's going to bind to the receptor on the plasma membrane of the liver cells that activates enzymes and releases glucose into the bloodstream. So it's part of your fight or flight. You're trying to get emergency energy. So in your liver, you're going to break down glycogen, and you're going to turn it into glucose. Your body's going to stop picking up glucose. So here's kind of the reception part of that, and the transduction, I guess, as well. So we get our epinephrine binding. That's our ligand. Activates the G protein. GTP comes in along with that process. Now our G protein is activated. That moves down the membrane, turns on adenylyl cyclase, which is going to activate cyclic AMP. That's going to carry the message to protein kinase A. That's going to do two things that are going to basically increase the amount of glucose that's in our blood that we could then turn into ATP, and we can use to run away from a lion or something like that. Opposite of that is going to be lipid-soluble hormones. So again, the difference lies in the fact that in a lipid-soluble example, this ligand hormone, so estrogen, can enter into the cell. In this case, the receptor is in the cytoplasm and not in the nucleus, but this hormone receptor complex can now move in to the nucleus and can manipulate expression. This, whatever vitellogenin is, whatever it does, it's a protein that can go do something. So again, lipid-soluble hormones come into the nucleus and alter things, where water-solubles have to stay where there's water. So hormones can do a lot. Like I said with epinephrine. Different target cells are going to respond differently to the hormones. There's the same signal. We might have different receptors for the same hormone that's going to cause something different to happen. We might have different second messengers that, again, is going to carry that message differently. Think back to when we played telephone. If you say something to one person and say the exact same thing to someone else, they might follow those directions differently. And then we also might have different proteins based upon which type of cell we have. Our genome might be methylated, might be moved around so that we can only express certain proteins. And also, hormones are conserved across families, so we'll often see a hormone in wolves and dogs, and they do something different. So if we look at this picture, it kind of shows kind of what I'm talking about here. If you notice here, we've got the same receptor. We got a beta receptor, but they have different intracellular proteins, so the transduction pathway is going to be different. In a liver cell, glycogen breaks down and glucose is released because it receives epinephrine. In a skeletal muscle cell, same receptor, we bind to epinephrine, and that causes our blood vessels to dilate. That should make sense. We need extra energy from our liver. We need to make ATP because we're running from a lion. Our skeletal muscles need extra blood, need extra oxygen so they can keep running. So it makes sense that they would want to dilate. And then over here in the intestinal blood vessel, so blood vessels, just in different places, different receptor, modified receptor. That causes the vessel to constrict. So you're going to have less blood in your intestines absorbing nutrients. Again, that's good because you're trying to run from a lion. So you don't need to digest. It doesn't matter if you digest the last meal. You just want to make sure it's not your last meal. So that's part one. We'll pick up with local regulators in part two.